Hello and welcome to BIOS Frontier Science. Today we're thrilled to welcome Nathan Gianneschi, Jacob and Rosaline Cohn, Professor of Chemistry and Material Science and Engineering and Biomedical Engineering at Northwestern University. Thank you once again for joining us.
Thank you.
To kick things off, Nathan, could you please give us a quick introduction to yourself?
Yeah, as you said, Nathan Gianneschi, I'm at Northwestern University in chemistry, material science and engineering, biomedical engineering, and pharmacology. My research group is quite broad. I have quite broad interests spanning polymer chemistry, biomaterial science, and a strong interest in the development of electron microscopy for studying nanomaterials in liquids. And so, yeah, we have this nice multidisciplinary group of Ph.D. students, research scientists, and postdocs at Northwestern working across those areas. And we also have an interest in translational approaches and have started a couple of companies to pursue those efforts as well. So yeah, I'm really looking forward to today and this afternoon and a bit of a conversation with you.
Awesome, and excited to jump into some of the stuff that your lab is working on. But before we get into that, one thing we like to ask our participants is, do you have any North Stars or common threads that tie your research or your work together?
Yeah, it's interesting. I thought about this recently. I think we, going back to when I was an undergraduate, I was taking biochemistry fairly early on. I'd sort of managed to get into some more like chemical biology and biochem type classes, as well as the chemistry courses I was taking at the time. And I'd become really interested in this interface between, and really at the edge of what was possible in synthetic chemistry by way of looking at mimicking proteins. So I was kind of learning about these two worlds and thinking that perhaps if I were lucky enough to have a research career, maybe we could connect them. And if I look back now, it seems like a common thread. I don't think it ever seems that way in the forward direction, right? When you're really living through the tape. But if I run the tape back, it does look that way. And I think the inspiration really, obviously from natural systems as a chemist is quite common, right? We sort of look at proteins and, the way they're capable of catalyzing reactions that still elude us, at least under conditions that elude us. But it wasn't so much specific, I think, around catalysis as sort of the structural elements. So for example, as a PhD student, we studied completely synthetic versions of allostery, so allosteric behavior of catalysts. These weren't protein in nature at all. They're made from completely different bonds, including organometallic systems. But the idea that what if we could use shape to control reactivity and not shape just of sort of a single sort of ligand domain, but the larger architecture of allowing substrates to enter and exit, for example, a catalyst. And so that kind of, I think, stemmed from thinking of those types of opportunities. How do I, again, sort of mimic the way proteins function, not specific function, but the way that they do it? And this kind of carried into my postdoctoral research and actually has heavily influenced what we do now. And one of our main sort of efforts now in using polymer science to mimic proteins, perhaps how they fold, maybe how they enter cells or how they interact with other cellular components. So I do think that's been there. And I think it's become increasingly maybe a self-fulfilling prophecy that's been sort of a guide for us in terms of at least what I've been interested in.
Awesome, thanks for sharing. And so one of the polymer science that your team is working on is developing nanomaterial drug delivery systems that you call enzyme-directed assembly of particle therapeutics, or ADAPT. Could you give us an overview of this technology?
Yeah, this was one of the early kind of areas we started working on when I started my lab. I started my group at UC San Diego in 2008, and we didn't necessarily set out specifically with how it looks now, but we were really interested in this idea of developing really materials that would respond to particularly like disease-associated signals, and particularly in this case, disease-associated signals in the form of enzymes. And so what do I mean by that? Well, there'd be all kinds of enzymes that would be expressed in specific tissues, maybe inside cells, maybe excreted, so they're on the outside of cells. That would be specific to that disease state. One of the attractive things about materials or sensors of any kind that respond to enzymatic processes is that they're self-amplifying. So You think of things like ELISA, right? ELISA is obviously an in vitro assay that relies on enzyme turnover of substrate to get to really high sensitivity in terms of detection. That's true for PCR too, right? It's an amplification tool using enzymatic reactions, in that case, polymerases, DNA polymerases. So we're interested in whether we could use that, but instead of an in vitro tool or an assay, do it for in vivo diagnostics and then ultimately for therapeutic delivery. So there's a whole set of enzymes that were candidates that continue to kind of fascinate us, and a whole range of things we haven't yet done. The class that we looked at first for this particular technology were proteases that are associated with inflammation, produced in all kinds of disease states. So in prevalent in tumors, one of the areas we've had a lot of interest in more recent years, say in the last 5-6 years, is in heart attack. So immediately following MI, this is true in humans and animal models as well. The heart, obviously goes into a process following the heart attack, ischemia and reperfusion, into a process of inflammation. And that proceeds really in an unstoppable way towards this sort of scar formation, and the heart, essentially the muscle ceasing to be a muscle, and therefore leading to heart failure. And so with a collaborator of mine at UC San Diego, Karen Chrisman, we developed this materials approach that we'd looked at initially in tumors, but now for heart attack, where the particles could be, very small, maybe even we've got some systems that are like this with even individual peptides, for example, that could be systemically injected. They would circulate all over the body. They're obviously being opsonized and cleared, so they end up cleared into sort of liver and spleen. And these are sort of the unwanted pathways. But in addition, they're able to pass out from the vasculature into the muscle tissue that's now damaged and inflamed. And so it's quite swollen. And so it's a really good candidate for nanomaterials. So a nanoparticle will be able to exit into the infarct within the heart. The heart tissue is normally very well sealed to the outside environment, doesn't let anything in. But right after the heart attack, within a few days, you've got this opportunity to get a nanoparticle in. Now, what happens normally is the heart's under pressure, it's pumping away, you get very little accumulation of a material like that. But in our case, on the other side, inside the infarct, there's such high enzymatic activity, our particles respond to that and spring open, and they make like a gel type material in the infarct site. And so we've been using that. So this is this sort of enzyme directed process for particle accumulation, for nanomaterial accumulation. And what we're now working on is some more advanced methods for using that approach, but to deliver small molecule drugs. So you imagine anti-inflammatories, factors that may reinforce a more favorable healing pathway, and maybe head off or perturb at least some of that negative remodeling that goes on after heart attack. So that was the type of thing that we'd been able to have some success with. And there are many other enzymes. So the concept is, if you could use an enzymatic process that's amplifying like that to target, instead of just a binding event where you've got like a binding constant, could you sort of actively accumulate something in a catalytic way? And that's sort of where that stemmed from.
Interesting. And so you touched on this a little bit earlier on and pretty briefly, but what types of molecules can you use this material to deliver? Are there any limitations to it?
So generally the way that we've published on it has generally been amenable to encapsulating small molecules. So we've done a little bit in cancer where we've encapsulated within a particle that's responsive like this. So the shell of the particle essentially is the enzyme believes, well, it is a substrate for the enzyme. So it sort of mimics the natural substrate of that enzyme, which in this case is the extracellular matrix. And then inside that shell is a hydrophobic drug. And so initially we were sort of somewhat limited to, and many drugs are like that, hydrophobic drugs that would then be released over time. And so that's sort of what it was immediately amenable to. We have some newer systems. We recently published on some injectable peptide-based gel type materials that can carry hydrophilic molecules. We didn't demonstrate that with a drug. We demonstrated it with a small molecule dye that enabled us to label the tissue. I mean, that's another way to think of it, as a diagnostic agent. But really, we've been in more recent times looking at peptide-based therapeutic delivery as well. And so these are usually, in the case of the ones we're interested in, hydrophilic kinds of drugs. and high molecular weight, which was a problem at first. So we're increasingly interested in those types of therapeutics on a range of different scaffolds that we might get to today. But that's kind of where we're going. So it's not infinitely generalizable. And as I said, the most of what we've done in the literature so far has been around small molecule hydrophobic drugs for that particular mechanism.
Interesting. And so why did you choose inflammatory enzymes as the trigger for this material as your first application?
Yeah, so as I said, I mean, enzymes are, to me, really attractive as things that you would like to sense. And you could view us like these materials as sensing the enzymes that are in the tissue because they're catalysts, right? If you're trying to detect a catalyst, you throw a substrate in and, it turns out one individual enzyme is going to turn over many, many copies. And as I said, that's the basis for some of the most sensitive detection methods we know, right? PCR, ELISA. So what if we could have a materials approach to use enzymes to amplify their accumulation or gelation within the tissue? Other kinds of things could be imagined potentially, but to us, enzymes and enzymatic action stands out for that reason. In MI, for example, those enzymes are highly upregulated. That's a very well-known initial response to all kinds of inflammation. One of the downsides in doing that is it's general. I mean, those proteases that are produced in that tissue are produced elsewhere. So if patients have other diseases that are related, so arthritis or other kinds of injuries also produce proteases. So there's potential for off-target effects because the substrates and the enzymes are not selective completely to those tissues. I've got a phone call. You might want to edit that bit. Unless you can't hear that. Hang on. Yep. Sorry about that. That they are they're not unique to those tissues. But the combination of, like I said, the porous nature now, the leakiness of the tissue, especially in the case of the heart attack, which again is true in the rat model, it's true in the pig models, it's true in humans. You've got, the leakiness combined with the enzymatic action, I think, has given us selectivity. And so it's a cool opportunity for nanomaterials because you've got, some selectivity from the fact that the nanomaterial can pass through this gap into the heart tissue, but not go elsewhere, other than, of course, some liver off-target effects. And the fact the enzymes are there, but that's why we chose those kinds of enzymes. High expression, well-known targets, well-known substrates, and potentially generalizable to other inflammatory disease. So not just MI, myocardial infarction, but maybe other kinds of disease states as well.
Interesting. And so you've talked about kind of using this in myocardial and then you guys have also explored it a little bit in tumors as well. But do you have any plans to move this forward towards a more commercializable therapeutic? And what indication would that be in? And what do you think that therapy would look like?
I think there's a strong opportunity in heart attack. And I think one of the reasons, one of the reasons we moved away from tumors, actually, we've moved for this system almost entirely away from cancer, is that the animal models are just less predictive of what happens in humans in the end. And that's not just for our materials, that's for all kinds of things, obviously, very famously. But for us in particular, the sort of physical features of the disease state kind of need to match, right? The leakiness of the tissue combined with enzyme expression. So because we've got these sort of two factors, I think you need to have something like this to be translatable. It needs to be in a model that makes sense, that translates. And these particular MI models translate quite well, at least in our opinion. And I think that's where, as I said, I collaborate with Karen Chrisman for many years on that. That was really her insight. She sort of said, why are you working on cancer? You should be doing this. And I think, we really believe that that's probably the direction to go in. One of the areas that we've been working hard on is to develop degradable versions of these materials so that they can be more translatable. So you can imagine more biocompatibility. The systems we work on now are they work well, but they're not degradable. They're not particularly amenable to that kind of process. So we're looking at that for sure. Another newer area for us has been looking at injuries associated with or like infections and things in the mouth. So I'm working with some dental kind of applications. And you might think of, in that case, there's an opportunity obviously for more local delivery. but using our system, maybe post-injection as a way to retain materials locally for sustained release of antibiotics and anti-inflammatories. And so that's also been an interesting direction that we're working with some people who are very translationally minded who have some excitement around that. And so these might have, interesting and unique applications where you want long retention, especially in difficult injuries or in patients with diabetes, for example. So there's a number of directions like that.
Makes sense. And so as you kind of further develop these nanomaterials, maybe as you start to get materials that are biodegradable, what other molecular or enzymatic triggers might you consider developing materials for? And what indications would you hope to expand into?
Yeah, so the as I said, I think there's the disease-related proteases that we've looked at. There's several flavors of these. There's some work on making substrates that are more and more selective. So there's the potential to make a more selective targeting agent that would maybe get around some of these problems I said of the fact that these proteases and proteases of their class are expressed in normal tissues, even like cartilage. So avoiding that and making them more selective is a push, as I said, degradability. But other enzymatic signals, we actually have had a lot of success in the recent few months on a completely different set of enzymes that are associated with the same disease state, but not generally involved in the inflammatory response. So these would be more selective because they're more specific to those tissues. That's literally something that I think a few of my collaborators and my students would kill me if I said specifically what target it is. But we're very interested in that for those reasons. Hopefully that kind of gives you the idea that, we'll see. We've said it's generalizable to other enzymes. We've never proven it in the literature, but we're just seeing it now, and we're excited to get that out in the world in the next few months.
Yeah, that'll be exciting to see. And so kind of as you continue in developing this technology, where do you think we'll be applying it 10 years from now? Or, where do you hope to see it being used in the future?
Look, I mean, I think all of us on this project really see it. We're fairly early on. I mean, it's an unusual way of developing a material or a therapeutic. It's targeted in the rat models. I think really the next step for us is to move with some of the more biocompatible materials into larger animal models. So I think the next 3 years is going to be really telling. If we have success in those kinds of systems, so pigs, for example, and good PK and all the rest of it, I think we really could have at least a shot at a translational tool. It's not an easy process, of course, to develop that, but that is definitely the goal. And I think in specifically in the MI applications where we have a shot. One of the interesting things that we think we have pathways to now is the delivery of some anti-inflammatory or inhibitors of inflammatory response pathways that have been shown in animal models at least to work quite well, but in humans, off-target effects created problems in clinical trials. So maybe we'd go back and look at some of those drugs, and that's kind of the classic nanomaterial, nanocarrier approach, right? We've got a way to target. Is that true? Okay, we think it is. Let's go back and look at where drugs would have worked if only for targeting. And so I think we're at that step now. If you get a good therapeutic effect, and as I said, it scales to the larger animals, then we've got a shot. And that's maybe not 10 years, but maybe more like half of that. So we're definitely pushing towards that now.
Oh, that's fantastic. But moving on to another project that your lab is working on. So in addition to your nanomaterial projects, you're also looking into a new approach to lipidation as a delivery mechanism for molecular therapeutics. Could you give us an overview of your ongoing projects here?
Yeah, so this was a project we started several years ago. This is actually in collaboration with, actually it came out of an industrial collaboration we had trying to take novel forms of lipids that are functionalized in a specific way, which I can describe, that would enable us to fairly easily modify small molecule drugs, peptides, oligonucleotides, again, through a lipidation strategy that's quite different actually to what had been done previously. And so the idea was, and the lipids being produced by this chemical process enabled them to be bifunctional. And so you would have a lipid that could grab onto human serum albumin in the bloodstream. And on the other end of the lipid, there's a functional group that would easily allow us to conjugate, as I said, small molecules, nucleic acids, peptides, whatever we want. And so what do I mean by this bifunctional nature? If you look at the way that, for example, albumin, which is the most abundant protein in our blood and that of other animals, of course, the way that it carries fats, which is one of its functions, is that fatty acids with these sort of polar head groups and hydrophobic tails stick their heads down inside albumin. So you're talking about 5 or so high affinity binding sites for things like stearic acid and myristic acid. And so they're like, if you imagine they're like snakes, they've got their head down these hydrophobic channels. And at the base of these hydrophobic channels within the protein is this little cup with a cationic charge and capable of hydrogen bonding basically with the lipid. And that's the way it's carried. In fact, other carrier proteins, so other fatty acid transporters also have the same kind of interactions. Hydrophobic channels with these little hydrophilic hydrogen bonding cationic type residues at the base of these hydrophobic channels. So these are features of common features of transportation of fats. What people had been traditionally doing in lipidation strategies is to take those fats, as I said, which are hydrophobic chains with this, anionic hydrophilic head group at the bottom. They've been take that linker and conjugate. So there are successful drugs that are done this way. For example, insulin-based drug therapeutics for long circulating half-life and things like that, where they've conjugated. That's my phone again here. They've conjugated the lipid to the molecule via that carboxylic acid, which is at the end of the fatty acid. And that kind of means that you've now got your protein of interest or a small molecule drug. Lots of people have done this with small molecules as well. And it's lipidated in the sense that you've now got a carbon chain, but that end group is gone. So in other words, it no longer looks like the native ligand for albumin anymore or other fat transporters. The technology that these guys came to us with was a way to keep that. So it's a molecule that looks exactly like, say, stearic acid, but it's got the carboxylate still there and another carboxylate. So I can bind my drug, but I keep the carboxylic acid. So now it can go and bind albumin. It can bind other fat transporters. And we showed that functions. So we showed it works really well in a case that we published for paclitaxel. So we took traditional, well-known cytotoxin paclitaxel, made that conjugate. We see extended half-life, so circulation time is part of it. But we also saw biasing towards tumor tissue that has upregulated fatty acid transport. So in other words, a lot of tumors are trying to consume more fats and proteins generally, right? So they're consuming these, the burger and fries, right, for the tumor to grow and continue to propagate. And we piggyback on that, or in other words, we hitchhike on that pathway by essentially disguising the drug as a fat, but in a really unique way, and a very subtle way, really, kind of an elegant solution because you've preserved those native binding modes. So in other words, albumin thinks it's carrying stearic acid, but it's actually stearic acid with a drug on the end. And that it then hands that off to the tissues, which also believe that they're consuming fats when in fact they're carrying the drug. So we call that like a Trojan horse kind of strategy, right? Which is a common expression used for that type of trickery, as you can imagine. And so we've been very interested in that for cancer, certainly, because you've got this bias targeting. In fact, Ozempic, you may have seen those ads on TV, semaglutide, a type 2 diabetes drug, uses that same fatty acid carboxylate to transport peptides. In that case, you're getting hundreds of hours of half-life from that, making that favorable contact with albumin, as opposed to a standard lipidation strategy, which might be tens of hours of half-life. So again, as I said, for what's out there in that case, it's a half-life extension. For us, or at least in our minds, that's what it was. For us, it was both that, but more importantly, I think it has particular power in cancer because you're able to leverage this rewired kind of metabolism where the cells are just consuming fat like crazy. So we can disguise ourselves that way.
Interesting. And so kind of just to clarify it real quickly is, your technology creates a more natural lipid that maintains that carboxylase.
Yep.
And so that has two effects. One, that you get, better binding to albumin. So that extends the half-life of any conjugated molecule. And then two, and this is something that, is this not seen with other lipidation approaches, this uptake into tumors?
Perhaps somebody has seen it. We don't. I don't know of an example, but there may be. What we see very specifically, and we've seen this through very specific inhibition studies, very specifically that is the mechanism. For example, we can block uptake of our drug by using selective inhibitors for that process. Mm-hmm. So what I will say is lipidation of small molecules is no secret, right? You bind to HSA, you get more extended of half-life. Potentially the drug becomes more lipophilic and you get better cell membrane uptake, but specifically pinning it to that process, like a more biased selective uptake process to do with those transporters, is uniquely enabled by this modification. So that lets us think about, how you might carry this forward. For example, we're excited about some new results recently that show that in certain cases, like for example, so patients who have been treated with antibody-based drugs like Herceptin, for example, can become resistant to those treatments. So these are patients with sort of HER2 positive cancers. They can become resistant to that treatment. It's been shown that part of that resistance, part of that pathway results in rewiring, metabolic rewiring, one of the outcomes of which is an upregulated fat transport mechanism. So you can imagine now you've got a cell migrating away from, say, the drug efficacy, so becoming resistant to the therapy, and then you've got a targeted cytotoxin that actually goes after that new pathway. So could you help those patients? Could you prevent those patients from going into a phase where they're resistant to the primary therapeutic? And so it's exciting, right? That there's a potential way of targeting or biasing the chemotherapeutic, but also maybe a way of helping patients who've met sort of one of these barriers of resistance. So that's kind of where we think we might be able to uniquely help.
Got it. And so, in terms of use cases and actual indications you guys are going into, is that what you guys are planning on tackling first, or are there other areas you're looking at as well?
That one, for this specific one, we have some issued patents around that molecule. So I think that's going to be that would be an exciting area to progress if we really think about clinical translation. We can conjugate all kinds of other molecules, and so our lab is interested in oligonucleotides, for example, and leveraging that same technology to get things like half-life. And you can imagine a whole lot of other small molecule therapeutics that may be interesting. But definitely in the cancer space, I think that's where at least I'm the most interested for that particular technology because of that, circulation half-life being a part, but then the bias targeting may be the other part of it.
So you actually just mentioned something interesting about kind of the oligonucleotides, nucleic acids. Do you think that this could be potentially used as a delivery mechanism for nucleic acids, like the new mRNA vaccines or gene therapies or things of that nature?
Yes, I think maybe and we'd shown some things sort of early on around half-life extension with this, and there are other people have worked on lipidation of oligonucleotides, some quite successfully, and to my mind anyway, it's well suited to maybe a siRNA type approach rather than larger transcripts, because you still have to get across membranes, which is always the big problem here. But yeah, I think being able to bias even siRNA, shorter oligonucleotides, sort of antisense applications, using something so simple, because you're not making a nanomaterial, you're just injecting this thing and it picks up native albumin. I think there's a lot of possibility there.
Yeah, definitely sounds like it. And it'll be exciting to see what you guys come up with in the future and how you guys progress this tumor-focused molecule or approach.
Yep.
But moving on to kind of the last area that your lab is working on, peptide therapeutics. Your lab is looking to a new approach to peptide therapeutics and have developed the protein-like polymer platform, or PLP. Before digging into the platform, could you give us a quick overview of peptide therapeutics and why it's an attractive space?
Yeah, so I think it's no secret that for many, many years, peptides, you know, probably if you look at different types of potential therapeutic modalities, they definitely pop out as one of the first things you discover, right? You look at protein structure, and fragments of proteins are, of course, peptides. If you look at hormones, right? We're just talking about diabetes, right? All kinds of agonists, antagonists of receptors with relevance in disease states, the sequences are known. They're coming from very, specific sequences associated with hormones that are naturally in our bloodstream, right? That we're naturally producing, and they're peptide based, right? So if you think about changing the course of a disease or mitigating it, maybe you take those hormones as a starting point, right? Or you take a protein crystal structure with a binding site where you can identify, a contact between two proteins that might be interesting to inhibit. If I can take one of the peptide sequences out of that, take that peptide, can it act to inhibit, say, a protein-protein interaction as an individual peptide? So these have kind of been the lead compounds, or one of the classes of lead compounds, or lead type, let's say, interactions that we then go on and modify in some way. And so we might modify it to such an extent that we get this peptidomimetic architecture, right? So in other words, a small molecule that mimics the original peptide. So we reduce its sequence, maybe we cyclize it, maybe we modify it very significantly, or we use that binding site to guide studies that would lead to a small molecule that may be nothing like the original peptide. But sort of a lot of things kind of connect to that. And if you look at, I think we looked at some statistics the other day about, certainly there's been an increase in approved peptide drugs, or at least peptides entering trials. But these are the very small percentage of actual therapeutics, right? You're looking at small molecules, increasingly, obviously, biologics, as in macromolecules, right? Antibodies and things like that. Peptides are sort of left out a bit. And there's lots of good reasons for that, right? They tend to have poor circulation half-life, that they're fairly small. They can be degraded easily, so that can be developed for. You can develop for some of these things, like increased half-life, degradation but these are generally things that have plagued them, in addition to things like you've got to optimize for that, and you've also got to get this peptide across cell membranes. So similarly, what we were saying about nucleic acids, these are molecules that are food, right? I mean, your body is trying to degrade them and turn them into amino acids, so it can go and make the proteins that it wants to make, not just take the protein you're delivering. So if you start thinking about, peptides as just a source of amino acids for your body, nucleic acids, a similar kind of thing, right? That your body is kind of in the business of rejecting artificial attempts to get you to take up things like that. And so the whole way along the line, if you have a peptide, for example, like I said, that's responsible for a specific interaction inside a cell, and I take that sequence, there's nothing inherent about that sequence that means it's going to get across the cell membrane. There's nothing inherent about it that means it won't have minute-long half-lives when I inject it. And so, as I said, over years, you can engineer a material that maybe still binds, but avoids some of those other problems. But it's difficult to kind of do all that at once. So you're sort of optimizing around multiple weaknesses. As that's our take on it. And so what if we took a completely different approach? What if we had a method that would let you do all those things, right? Maybe increase binding affinity while it's doing it. And so we really, in our lab, if I go back in time a little bit, we were very interested and actually it came from some of our enzyme responsive kind of work that I'd mentioned earlier. We were looking at ways of displaying peptides on polymer scaffolds where we would retain some of the biological function of those peptides and use the peptides to kind of guide, say, a polymer. The polymer may be a carrier of a small molecule drug, and that's kind of what we were talking about earlier. One of the things we realized when we displayed peptides on a polymer backbone in very high density, so in other words, every single side chain of that polymer is a peptide. So there are no spaces. We were able to do that because of really advancements in living polymerization chemistry. So we're sort of leveraging fairly modern methods and tools of various kinds, radical polymerizations, metal initiated or metal complex initiated reactions as well, sort of leveraging several of these under mild conditions to make architectures where we could take arbitrary peptides, lots of different peptide sequences, not just one or two. But if you've got a drug that you'd like to develop, you can take that peptide sequence without modifying it and display it on this scaffold. And one of the first things we noticed was that we got proteolytic resistance immediately. Almost universally, this is true. We can engineer some other elements, but basically the peptides are protected from degradation. And what we realized through some structural analysis is that the polymers, should be like a linear chain with peptides coming off it. It actually globularizes. It forms like a little globular structure with a hydrophobic polymer in the core, and it's kind of wrapped in peptide. And that structure turns out to be really important. So, this sort of emergent property comes up, right, where we didn't necessarily design it that way. We were trying to do something different, make a protease substrate. Proteases won't cut these things anymore. So we started looking at other features. Does it get in cells? Like if we make them cell penetrating, will they get in the cells? The answer is yes. Do they get in the cytosol? Can we inhibit protein-protein interactions? Or can we address, for example, organelles inside cells? Can we essentially not just be trapped in endosomes? And the answer to that was yes. Because we've got multiple copies of peptides on each polymer, is it multivalent in binding? And yes, they're cooperative. They bind in the way you would expect. So in other words, the whole is bigger than the sum of its parts, right? And so you're getting these sorts of properties, and we've become really interested in this because now with molecular weight control, you're going in this other direction, right? Instead of taking a peptide and taking a small molecule sort of chemical biology approach to then turn that into a drug, which is say a peptide goes to a peptidomimetic or a small molecule, we're going the other way. So we're taking that lead peptide or an incretin, a hormone straight off the shelf, and making it into a larger and larger architecture, a multivalent protein-like architecture. That's why we call them protein-like polymers. And so these have features of proteins, physical, physicochemical, the way they actually behave. I mean, they do simple things like run on electrophoretic gels, the same way as the protein ladders. We can characterize them by some of those tools and purify them by those methods. We can put affinity tags on them, all the kinds of things you think about in protein engineering. But you're on a material that takes us a few weeks to make, and we can make many variants of it and then check and compare these. So we've got projects in the group, for example, that look at all of those things, peptides that might have come from phage display between, maybe crystal structures and informed by crystal structures, and then purely hormone peptides for sort of achieving those goals, long circulation half-life. You're talking about week-long circulation half-life by displaying like this, blood-brain barrier penetration, cell uptake, and there's sort of emergent properties again from this initial serendipitous discovery that we were led to because we were trying to again sort of get peptide function, enforce it on the polymer, and then use this in a way to do therapeutic delivery. Now and then through all of that quickly realized what we were really doing was delivering the peptides, but as sort of this cooperative entity. So it became about not a delivery system, but actually a way of organizing peptides to mimic the way that they're organized around proteins. Sort of dynamic enough to be able to bind targets, but restricted in motion enough to not be just food for proteases. And that kind of has led to this sort of new area for us, which has been really fun and really exciting to bring in biology collaborators and really get more into the clinical translation.
Interesting. And so this platform has obviously addressed a lot of the shortcomings that has inhibited peptides and their use as therapeutics. Do you guys have any applications for this technology that you're exploring? Or if not, are there any interesting applications you would like to explore using the PLP platform? Yeah,
so like you said, I think that this has sort of led us, and I was just saying, the biology groups we talked to a lot of MDs. We've got people who are experts in specific biochemical pathways and have, years-long experience with, wanting, all of the tools there to sort of look at sort of famous pathways that have led to neurodegenerative disease or cancer, for example, to be able to get into a cell with a material that's able to inhibit, say, protein-protein interactions or amyloid fiber formation, right? If you think about Alzheimer's and things like that. if you've got a multivalent way of doing that with peptide leads, you sort of generate a lot of excitement around that, right? So it's kind of thinking about it, and we sort of have thought about this as these are really material science type approaches to thinking about those kind of biological interactions, which have really been the domain of the field that we call chemical biology. But a lot of the outcomes of these things, these protein interactions that go awry, are materials, right? They make fibers or protofibrils and other architectures or multi-protein complexes. And so intervening in that has meant that we've been able to say to folks, oh, we have a way to sort of size match with, in other words, really mimic a specific protein within a, say, a dimer pair. And because of that, we've been able to look at several different targets. So we're looking at neurodegenerative disease, as I kind of sort of mentioned, at cancer. We're interested in, and those are all systemic injectables meant for delivery, if you like, of that peptide on target. We've got some, as I mentioned earlier, data on our ability to penetrate the blood-brain barrier. And these are functions of long circulation, so multiple day circulation, half-life plus some design features that let us do that. And to me, that's kind of one of the holy grails, right? A systemic injectable peptide that can get inside cells and perturb those interactions. And so, you could do 1,000 projects. I think we have maybe tens of projects, but they really, the ones we're most excited about is where you can't do this with another therapeutic modality. So inside a cell and an interaction that's been, dubbed undruggable. Certainly those interactions are being addressable with peptides through different approaches, but I think scaling it and getting the right sort of performance, let's say, in animals and ultimately in humans is going to be key. There are other areas too, right? You think of more local administration in the case of ophthalmological targets that have been really interesting to us. And some of those are extracellular. We're interested, for example, in, as I said earlier, in taking hormones, right? So you're looking at some extracellular targets there as well, and whether or not the polymers kind of alter, right, the way that hormones behave. So do you get different kinds of effects by the way that you immobilize them and organize them in 3 dimensions? And so without, I guess I didn't give you specifics, but that gives you an idea of the scope of the type of thing we're doing.
Yeah, no, those are all very interesting projects. I guess, though, would love to hear kind of specifically, what do you think, are there any that you think are the most promising, or any that you would like to see move forward over the next couple of years?
I think, if we think of, and I just not even speaking to, whether, the commercial side of this, which is always, or what's sort of the most interesting to sort of other people or anything like that. I think one of the most sort of, if you think of a new drug modality and where an area that's been crying out for this, I think is a neurodegenerative disease. You look at, problems that have really recently, this is very topical, right, have kind of plagued that field. Of course, it's complicated, right? It's the way that clinical trials are done and other things. But if you've got a material that can enter a cell and engage these targets in a completely new way, then you have a new therapeutic modality. And ultimately, the demonstrating that works opens all kinds of other doors for other kinds of disease states where similar protein dysregulation is responsible for the disease. And those are really protein-driven diseases, right? And the difficult interactions within the cell to engage selectively unless you, they're just difficult generally. And so I think that's a high bar. And I think that, for a new sort of modality like that, you want to be able to jump over, I think, a high bar in order to be able to demonstrate that certainly it's going to be differentiated from what you could do with an antibody, for example. There's all sorts of, I think, good arguments for why you may want to develop these ultimately instead of antibodies for different kinds of receptors, even if you could develop an antibody for it. And that may include things like tailorability. We have a lot of control over molecular weight, functionalization, non-natural amino acids and other modifications you may want to make in order to tweak affinity. So there's ultimately that kind of flexibility you think of when you think of small molecule drug development, but sort of on this other kind of scaffold. And so I think that to me is the area where, again, very high bar, but you think about systemic administration for those types of diseases, and you're getting at something that's currently an unsolved problem. And I think ultimately that's where you really need to be.
Yeah, that'd be fantastic, and definitely a huge unmet need right now, particularly with the recent spat of drug failures and clinical trials that you mentioned.
Right.
Awesome. Well, we are kind of running close to the end of our time, but before we go, I have a few last questions for you. First off, where can people learn more about your work?
So the easiest route is through our website, which I could provide to you if you wanted to have it on the screen or something like that.
Yeah, we
But
can
I'll
link to your website in the description.
link to the website. Yep. Our group website has all of our most recent papers, some news articles on these kinds of topics, and some other links to sort of the broader area. I think that's probably the best port of call. Yeah, does that make sense?
Yeah, absolutely. Awesome. And before we go, do you have any shameless plugs or last closing thoughts?
No, thanks for having me today. I think, you actually covered a whole lot of stuff that we work on. I'd say by shameless plug, I mean, we do work in other areas. We're actually developing some melanin mimetic materials that mimic the melanin pigment that we have in our skin and eye and hair. Like all animals on Earth, almost all organisms actually use melanin for that purpose. We're developing it for some cosmetic and skin applications. We've got some papers out there, and people can see more on that on our website as well. That's an area that I think is also ready for translation. And so we'd be very interested in follow-ups on that as well. I like the word shameless. I think that pretty much is it. And thanks for having me.
Awesome. Well, thank you so much for joining us, and thank you for an absolutely incredible episode. This was a lot of fun, and we're really grateful that you took the time to do this.
Yep, me too. Thanks a lot.